In this video, we can discuss about the identifications of microorganisms. It's an important topic of Pharmaceutical Microbiology Unit 2 of 3rd semester B-Pharm. There are different methods for the bacterial identifications like microscopic morphology, macroscopic morphology, physiological or biochemical characteristic, serological analysis, pathogenicity test, and genetical and molecular analysis like guanine cytosine base composition, DNA analysis using genetic probes, nucleic acid sequence, and rRNA analysis. So in this topic we have to discuss about the microscopic morphologic methods as well as physiological and biochemical characteristics. So coming to the first method, microscopic morphological method, we can study the microscopic morphology by using different type of microscope like light microscope or electron microscope, which we had discussed in detail in unit 1. Now by microscopic morphological methods, we can identify the shape and size of a microorganism and Gramm staining reactions of a microorganism, acid-fast reactions of a microorganism, and we can identify some special structures of a microorganism. So coming to the different shape of bacteria, which we have already discussed in unit 1, microorganisms can be classified according to their shape like coccus, bacillus, coccobacillus, vibrio, spirillum, spirochete, scoies, and star. Now according to the flagellar arrangement, microorganism can be classified as monotrichous, lophotrichous, amphitrichous, and peritrichous. Now in microscopic method, the microorganism can be studied by different methods like hanging drop method, wet unstained film, or by stained film. So in hanging drop method, the microorganism in natural state are best viewed when suspended in a clear fluid. So in this we will make the drop to hang on cover slip so that we can easily study the microscopic morphological structures of an microorganism, especially the motile microorganism. Now second one is wet unstained film. In this method, the drop of liquid specimen are placed on a slide and covered with a cover slip and examined with high powered dry objectives. So it is a direct method without staining. So third one is stained film. A drop of sample is smeared on a glass slide and dried in air, then passed over through the flame to fix it, then fluided it with appropriate dye solution and fix it and observed in the microscope. It is the routine procedure in microbiology. Now coming to the important part of this topic by microscopic morphology, stain and staining techniques. It's an important topic from examination point of view, both practical as well as theory examination. Question has seen as define and classify stains, explain about staining techniques, and in practical examination the major experiment will be the perform the Gramm staining procedure and identify the microorganism, as well as study the motile activity of a motile microorganism by hanging drop method. What is staining? Staining is an procedure for artificial coloration of microorganism with a dye that emphasizes certain structure to produce the color contrast. So that is the staining. Now what is smear? Smear is a thin film of solution of microorganism on a solid. Only the suspension of microorganism. We are not added the stain, and after fixing only we will add the stain. So the smear is usually fixed to attach the microorganism to the slide and to kill the microbes. And the microorganisms are fixed and stained to increase their visibility and to make more noticeable the morphological structure like flagellae, and to preserve them for the future study, to develop the contrast with the surrounding. So these are the reason for fixing as well as staining. Now coming to the stain or dye. So stain is defined as an organic compound which containing two parts, chromophore and auxochrome. Chromophore is a group with conjugated double bond and that will give dye its color. So this is the part of the stain which will give the color to the stain, that is chromophore. Now auxochrome, these are the part which will bind to the tissue, which is these are the group linked to the benzene ring by ionic, covalent or hydrophobic bond with binding to the tissue of organism which is to be stained. The stain or dye can be classified according to their ionizing nature into two, that is acidic dye and basic dye. Now what is acidic dye? In acidic dye, the chromophore, the part of the stain which will give the color, will be an anion, which on ionization it will produce an anion. It is negatively charged. It possesses negatively charged group like COO minus and OH minus, and this part will bind with positively charged structure and it will repel the negatively charged cells. So examples of acidic dyes are rose Bengal stain, Congo red, eosin and acid fusion. So these are the example for acidic dye. In this, chromophore is anion which is negatively charged, which will repel the negatively charged cell. Now coming to the basic, in this the chromophore, the color giving part is cation which is positively charged. It possesses positively charged groups and it will bind with the negatively charged molecules like nucleic acid and other many proteins. Since the bacterial cell are also negatively charged to their composition of their cell wall, this basic dye will bind with the negatively charged cell. So this is most commonly used stain because there will be an attraction between basic dye as well as the negatively charged cell. So examples of basic dye are methylene blue, basic fusion, crystal violet, saffronin and malachite green. Now along with the stain we may use some other component like intensifiers as well as mordant. So intensifiers are a physical or chemical agent which is used to increase the intensity of stain. It will increase the color of the stain, that's all. Now coming to the mordant, it is defined as any substance that form an insoluble complex with the stain and serve to fix the color to the bacterial cell. This main intention of using mordant is to fix the bacterial cell with the color. So mordant will increase the affinity of stain toward the cell component, and mordant can be applied in three ways. Either pre-mordanting, in this mordant are applied prior to the application of stain, then meta-mordanting, in which in conjugation with the stain, and post-mordanting, in which mordant are used after the application of stain. So that is the mordant. An example for mordant is Gramm iodine which is used in Gramm staining. Now coming to the fixation. Fixation is a process by which the internal and external structure of cell are preserved and fixed in position. Microorganism is either killed or firmly attached to the slide during the fixation. It will inactivate the enzyme that may disrupt the cell morphology, toughen the cell structure so that they do not change during the staining and observation. And there are two types of fixation, either by heat fixation or by chemical fixation. In heat fixation, it is done by gentle flame heating of an air-dried film of bacteria so that it will preserve the overall structure but do not fix the intracellular structure. In case of chemical fixation, we will use some chemicals like ethanol, acetic acid, mercury chloride, formaldehyde or glutaraldehyde. It is mainly used to protect the cellular structure as well as substructure and morphology of bacteria. The fixatives will penetrate to the cell and it will react with the cellular component and render them immobile. Now coming to another part of this topic, staining techniques. Previously we had discussed about the stain, classification of stains, examples of stains. Now coming to the staining techniques. So it is mainly classified into the simple staining, differential staining, special staining, staining of specific structures and staining for electron microscope. Now coming to the simple staining, in which we will use a single stain. Usually that will be a basic type and it will provide color contrast and also it will impart same color to the all bacteria. Whatever be the color of the stain, that will give to all the bacteria. And this method, simple staining method, is used to study the shape and also to count the number of bacteria. Examples are methylene blue, crystal violet or basic fusion. Any one of these basic stain are usually used to stain in simple staining. A mordant also can be used to hold the stain or to coat the specimen to enlarge or emphasize its structure. Now, difference between the differential staining is, in this simple staining we will use only one stain. In differential staining more than one stain. So in simple staining it will give only one color to all the bacterial cell. Differential stain will impart two or more different color to bacterial cell. And simple staining reveal the size, shape, arrangement of bacterial cell, and along with that in differential staining it will give an additional, it will differentiate the microorganism to the two group of bacteria. Now example is methylene blue staining, and for example for differential staining is Gramm staining and acid fast staining method. Differential staining, in which we will differentiate the bacteria to the separate group based on the staining property. There are two types of differential staining, Gramm staining and acid fast staining. Now coming to the Gramm staining, it's an important topic which has seen as a 5-mark question in theory, as well as it is the major experiment of practical microbiology. Gramm staining is devised by Christine Gramm in 1884. In this, we can classify the microorganism into Gram positive and Gram negative microorganism. Gram positive bacteria which will tend to kill by penicillin and detergents, and in case of Gram negative bacteria which are more resistant to antibiotics. Now this is the procedure for an Gramm staining. Here we will use a primary stain. We can use different stain like methylene violet, gentian violet and crystal violet. Crystal violet is the commonly used stain and we have to treat for 1 minute. Then we will use a mordant by using Gramm iodine again for 1 minute. Then we will use a decolorizing agent. Usually it is an alcohol or ethanol. We can use acetone or aniline also for 10 to 30 seconds, and counter staining by using safranin. So this is the procedure. We have to clean the slide. After that we have to air dry the slide to dry it, and then we will prepare a smear of microorganism on the center of the slide, and then we will add the primary stain crystal violet usually for 1 minute. After that, after the washing, we will add the mordant Gramm iodine. Then after again washing, we will add decolorizing agent alcohol ethanol 95%. Then after washing, we will add the safranin. Now let's see what will happen during the Gram staining reaction. So while adding crystal violet, both gram positive and gram negative bacteria will take the color of crystal violet, that is purple or blue. While adding gram iodine, that is used as a modern, it will impart the same color for gram positive as well as gram negative bacteria by forming a complex with the primary stain crystal violet. Now as decolorizing agent we will use alcohol. That time the gram positive bacteria will resist the decolorization and it will retain the color of primary stain. So in gram positive bacteria that will resist the decolorization and it will give the color of primary stain, that is the color of crystal violet. But in case of gram negative bacteria, it will decolorize the color of primary stain to colorless, and finally while adding the counter stain or basic stain safranin, which is a red color, the gram positive bacteria will show the color of primary stain, that is purple or blue, and gram negative bacteria will show the color of secondary stain, that red. So according to the color of microorganism, we will differentiate the microorganism into gram positive bacteria and gram negative bacteria. So the blue colored organism will be gram positive bacteria and red colored organism will be gram negative bacteria. So what is the reaction? What is the reason for this one? So we know that while adding the crystal violet, the primary stain and modern will trap inside the cell wall of gram positive bacteria due to its complex nature or thick peptidoglycan layer. So it will remain inside the peptidoglycan layer as it has already formed the crystal violet gram iodine complex. So it is very difficult to decolorize, or it is very difficult to remove from inside of an gram positive bacterial cell. So it will retain the color of primary stain and there is no space for to get into the cell in case of gram positive bacteria for counter stain. But in case of gram negative bacteria, due to thin peptidoglycan layer and more porosity due to the poring channels, what will happen during the decolorization stage, the complex crystal violet iodine complex will easily diffuse back to outside the cell. So while applying the counter stain, its color will shown by gram negative cell. So this is a micrograph of a gram stained bacteria. The blue colored organism is gram positive. Blue colored organism may be either bacillus or cocci. Anyway, the color will decide the gram positive or gram negative. So blue color will be gram positive organisms and gram negative will produce a red or pink color. This is another micrograph. Here blue colored organism show gram positive cocci. Here gram negative bacillus. These are some examples for gram positive and gram negative bacteria. Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pneumoniae, Corynebacterium diphtheriae, Clostridium botulinum, Bacillus anthracis and Clostridium tetani are examples for gram positive organism. E. coli, Salmonella typhi, Vibrio cholerae, Neisseria gonorrhea, Klebsiella pneumonia, Haemophila influenza, Bordetella pertussis are example for gram negative organism. Now coming to the mechanism of gram reaction. Even though we are using same stain, same primary stain crystal violet, same modern gram iodine and same decolorizing agent ethanol and same counter stain safranin, gram positive bacteria will show blue color and gram negative bacteria will show pink color. This is the reason that gram positive bacteria have more acidic protoplasm and reduced porosity of due to the thick peptidoglycan layer, so that they will retain the basic dye or basic dye iodine complex to greater extent and it will resist the decolorization. But in case of gram-negative bacteria, due to the thin peptidoglycan layer and discontinuity in outer membrane structure and more porosity due to the presence of porine protein, the crystal violet or crystal violet iodine complex will readily remove during the decolorization step. So by applying the counter stain, it will produce the color of counter stain, that is pink. So this is the mechanism of Gramm reaction. So here let's see, it will produce a complex while due to the thick peptidoglycan layer, it will produce dimodant complex which will resist the decolorization. But in case of gram-negative bacteria, due to the thin peptidoglycan layer, it will easily decolorized by decolorizing agent, and finally it will produce the color of secondary stain or saffronin. So there is some exceptions, like if we are decolorizing for a prolonged time, even gram-positive bacteria may show the gram-negative result. Inadequate decolorization may cause all the cell as gram-positive bacteria. So if we are reducing the time of treating decolorizing agent, even gram-negative bacteria also will show gram-positive result. So the modern also increase the interaction between the cell and dye. Removal of cell wall with lysosome from a gram-positive bacteria result in gram-negative staining profile. So this is the basic structure of gram-positive and gram-negative cell wall. Here it is a thick peptidoglycan layer is there. Here a thin peptidoglycan layer and outer membrane is there. And outer membrane is more porous due to the presence of porine proteins. Gram-positive and gram-negative bacterial cell differ in the following characteristic. In staining, it will give purple color for gram-positive and pink color for gram-negative. And there is difference in structure of cell wall. We have already discussed in previous chapter. There is a difference in growth requirement for gram-positive and gram-negative. And there is a difference for susceptibility to antibiotics. And pathogenicity also different in case of gram-positive and gram-negative. Next differential staining method is by acid-fast staining. So it was discovered by Enrich and modified by Zeil and Nielsen. So acid-fast staining technique is also known as Zeil-Nielsen methods. This method is mainly used to identify Mycobacterium species like Mycobacterium tuberculosis and Mycobacterium leprae and Nocardia species. So this is a preliminary test for the detection of tuberculosis. In this, we can differentiate the microorganism into acid-fast microorganism and non-acid-fast microorganism. So the property of a cell to retain the basic stain or the primary stain in presence of acidic alcohol is known as acid fastness, and the organism which showing that property is known as acid fast microorganism, and the microorganism which lose the basic stain during acid alcohol decolorization is known as non-acid fast bacteria. So the main property for the retaining or resistance towards the decolorization is due to the presence of waxy cell wall which containing mycolic acid. So the cell which having this mycolic acid will resist the decolorization and which will show the color of primary stain, and that is known as acid fast bacteria. Now coming to the procedure, we will use primary stain cardboard fission for 5 minutes. After that, we will use decolorizing agent acid alcohol for 30 to 40 seconds. Then counter stain methylene blue for 30 seconds. So this will be the reaction towards acid fast staining. Acid fast bacteria will retain the color of primary stain till the last step. So it will show the red color, and non-acid fast bacteria that will show red color during the primary staining and during decolorization it will decolorize and it will be colorless, and during counter stain it will produce a blue color. So finally the acid fast bacteria will show red color and non-acid fast bacteria will show blue color. So this is a procedure. So here we will take the sputum smear for a tuberculosis patient and we will fix it on an air dried slide. Then we will add carbon fission and we will heat gently for 5 to 10 minutes. Look for the green scum, and we should take precaution to do not dry it. After washing we will add acid alcohol, and for decolorization, after decolorization we will add the counter stain methylene blue. Again we will wash it and we will go for oil emulsion objective lens. So that is the procedure. Step 1, heating with basic fusion and phenol, that is the mixture of basic fusion with phenol is known as carbon fusion, and then by decolorization with 20% sulfuric acid, or then counter stain with methylene blue. So this is a micrograph of acid fast staining. Now coming to the next staining for special characterized negative staining, or also known as capsule staining. In this we will use Indian ink and nigrosin, and this is mainly useful for the demonstration of bacterial capsule which are seen outside the cell wall. So usually which will not take the stain, and it can be also used for the demonstrating slender spirochetes. So we will get an uniformly colored background against which the stained bacteria stay in contrast. Very slender bacteria such as spirochete are not demonstrable by simple staining method, can be viewed by negative staining. So this will be the observation of an negative staining. We will get an dark background and a colorless microorganism which will show the capsule or negative staining. Another micrograph of capsule staining. Next one is spore staining. In order to stain the endospore, Bacillus and Clostridium form endospore, which are a resistant structure to survive the unfavorable environment. So endospore are not stained well by dye, but once the stain they will resist the decolorization. So it can be done by Schaffer's Filton method, in which we have to eat the bacteria with malachite green and then washing with water, then counter stain with safranin, so that we will get a green endospore in a pink to red vegetative cell. So we can see the observation as pink vegetative cell and a green endospore. Now next staining is flagellar staining. In order to stain the flagella, bacterial flagella are thread-like organelles of locomotion that are also slender, which can be directly seen only in electron microscope. To see in light microscope, the thickness of flagella is increased by coating with tannic acid and potassium alum and then stained with para-rosanilin in case of Lifson method and basic fusion in case of Gray method, so that we will get an observation like this. It will be stained in the flagella. Now impregnation method. This method of staining is used for the cell and structure that are too thin to be viewed under the ordinary microscope, so that we will impregnate with the silver on the surface. It is used to demonstrate spirochetes and bacterial flagellae. Next staining method is fluorescent staining. In this, ultraviolet light is used to excite the molecules so that they will release the light of longer wavelength than that of originally striking them. So they will use the dye that emit the visible light when bombarded with short UV rays. These pigment is also called as fluorochromes, example like oromin and acridine. The different wavelengths produced are often seen as brilliant shades of orange, yellow or yellow-green. Now these are some questions seen from this portion. Write a short note on staining techniques. In that we have to write simple staining, differential staining and special staining for special characters. Write a short note on Gramm staining. We have to write the principle, procedure, mechanism of Gramm staining, and write a short note on acid-fast staining. In that we have to write the principle, procedure and mechanism of acid-fast staining. And define and classify stain. We have to define stain and we have to enlist the different parts of stain, Auxochrome and Chromophore, and we have to classify the stain, acidic stain and basic stain with example. Now principle and procedure for Gramm staining for 5 marks. Thank you for watching this video.